I'm going to pause here and give a moment and actually stop my screen share to give a moment to introduce our other friends on the call, which would be Pietro and also Caroline. So do you want to give yourselves a quick intro?
Cool. I'll jump
Sorry, I
in
didn't tell you who
first and
first.
then no
Go for it.
worries. I'll jump in first and then pass the mic to Pietro. So I'm Caroline. I'm on the Human Computation Institute team. So I get to help out with stall catchers and other projects. And today's particular project is really webinar is focused on stall catchers. Our colleague Maggie Lane is also helping a lot behind the scenes with stall catchers. So you might correspond with her in the stall catchers chat, perhaps as you're participating. But we are lucky to have Pietro here, the director of the Human Computation Institute that created stall catchers. So he'll tell you more about himself and the project momentarily. So Pietro, I'll pass the mic to you.
Thanks, Caroline. Yeah, it's appreciate everyone showing up today for this. It's I always feel a sense of great gratitude for everyone who participates in stall catchers, and obviously the goal is to help advance Alzheimer's disease research. So it's kind of one for all and all for one, and so just want to say thanks. So I am involved with the project. I started the project with a bunch of other people. Thinking first meeting that led to this was probably way back in 2013, and around 2014, I pitched the idea at the National Alzheimer's Summit at NIH, and people got interested, and eventually that led to some funding support that allowed us to create the project in earnest. But I work closely with Chris Schaefer and Nozomi Nishimura, the biomedical engineering lab at Cornell, who are doing the actual biomedical research. So I don't have a background in biomedical research at all. I'm a cognitive scientist, and so my background is actually in thinking systems, whether they're natural or artificial, which of course is becoming more interesting today with all the AI stuff that we see. But the question I've always been curious about is, are there ways we can combine the sort of the complementary strengths of humans and machine-based systems to solve problems that can't be solved by humans alone or by machines alone? And so stall catchers is kind of a simple version of this, where the machine-based processes do a lot of the background work to coordinate taking a lot of data and then parceling out little bits of it to different people so we can all work on it, and then collecting everything back together to provide a final analysis back to the Cornell lab. And each data set in stall catchers is evaluating a different either a candidate treatment for Alzheimer's or something that would help us better understand what a candidate treatment might look like. So that's kind of a rough overview.
Awesome. Thank you so much. All right, we do have some descriptive slides for us to explain what stall catchers is for anyone who's new to it. And for that, I can move forward, or if Caroline or Pietro want to take over as the more expert version of this, since we have you. Pietro, can I just throw it back to you to give
Oh,
some?
yeah, sure. So as I was saying, I met Chris Schaefer, who's a professor in the biomedical engineering lab, about over 10 years ago now. And we were introduced by a mutual colleague who was in citizen science at the time, Janice Dickinson, at the Cornell Lab of Ornithology, which is famous for the eBird Citizen Science Project, although her project was called Yard Map, a different one. And I think that eventually got a new name when it was adopted by the Audubon Society, that I can't remember now. But anyway, that's just a little kind of citizen science history. So anyway, Janice introduced me to Chris, and then I said, "So what are you working on?" And he said, "Well, we're studying Alzheimer's disease," and of course I knew nothing about it. I mean, except what most people know. It's a form of dementia. And he said, "Yeah," he said, "there's this aspect of the disease that's long been known but never understood, which is that in patients with Alzheimer's, particularly in the more advanced stages, there's 30% less blood flow in the brain." And to get a sense of that, that's kind of like the feeling you get if you've been lying down for a while on the couch and you stand up suddenly and you feel sort of woozy. That's because you have about 30% less oxygen in your brain. It takes a minute for your heart to pump that blood back up to your brain. But that's the state of the brain in a lot of Alzheimer's patients. And so Chris and Nozomi, who run the Cornell lab, were curious about whether investigating what's causing this reduced brain blood flow might lead to potential treatment possibilities. And and they made a really important discovery, which is maybe next slide. We'll see what it looks like. Haven't looked at these particular slides. Yeah, perfect. So they were looking at the tiniest blood vessels in the brain, and they were looking at what we call mouse models of Alzheimer's disease. So that's sort of a fancy way of saying that they have mice that were given the human gene for Alzheimer's disease and bred with this gene, so they manifest Alzheimer's, the symptoms of the disease, and all the kind of physiological characteristics like the amyloid plaques and so on. So it's considered, you know, there's always the question, is an animal model of a disease really exactly the same as a human model? It's not, but we can still learn a lot from these animal models. And the mouse models of Alzheimer's have been actually very helpful to advancing our understanding of the disease. So anyway, they were looking at blood flowing through the tiniest capillaries of the mouse brain, curious to see if that's where there were some obstructions. And it turns out that the Alzheimer's mice did have about, I think, 4 times as many stalled capillaries in their brains compared to healthy mice. And the interesting thing is that when they did an intervention, it was kind of an accident. Actually, it's what we call a serendipitous discovery, where you do one thing for one purpose and then suddenly it helps you, you know, it teaches you something you weren't expecting to learn. So they were trying to do better imaging on these mouse brains. So they injected something into the mice to make them light up better under the microscope. And it turns out that whatever they injected actually made the stalls go away in the mouse brains, and their blood started flowing again, and then they also noticed that the mice were getting their memories back when this happened. So this was a hugely exciting discovery. The problem was that what they were injecting the mice with was something you could only do very occasionally without harming the mouse, and it would be the same for a human. So if you were constantly doing that to a human or a mouse as a treatment for Alzheimer's, the treatment would actually be worse than the disease. It would, you know, you wouldn't be able to survive it. So the plan was, okay, now we understand that capillaries are stalling in the Alzheimer's brain, and that if we can get blood flowing again, we actually get an abatement of these cognitive symptoms. So how do we safely do this? Can we find a treatment that is sustainable, that could do this? And that's what Stall Catchers is all about, trying to kind of narrow down possible treatment approaches that could actually restore brain blood flow, you know, without harming the mouse or the human. So next slide.
If I could click it, there we go.
There we go. So I thought, you know, wow, this sounds really exciting and promising, and I really wanted to help any way I could, but I wasn't sure how I could possibly help. And I said, so why do you think we're talking about this? And Chris said, well, we have this kind of analytic bottleneck where when we do an imaging session and we you know, look in the brains of all the mice that are part of this study for a particular experiment, it takes about a week in the lab to do the imaging, but then it takes 6 to 12 months to analyze all the data, because we have to go and look at every tiny little capillary in the brain and figure out whether it's flowing or stalled and count the stalls to see whether or not some kind of treatment we're trying is actually reducing the stall rate. And it's just one of these things that machines don't do very well. And so he said, we've tried using machine learning and other kinds of machine-based techniques, and he says it's only about 85% accurate, and our tolerances are much tighter than that. We need to have at least 95% accuracy 95% of the time. So I said, oh, okay, this sounds interesting. You know, maybe we can figure out a way to crowdsource this through citizen science. So next slide, please. I won't give all the details, but with a lot of help from a lot of people, we figured out a way to gamify the task of analyzing stalls in the brain, and that ended up becoming the Stall Catchers Citizen Science Project. Next slide. So today, and literally today, we have 140,000 people who have done the Stall Catchers activity. So we just hit this milestone. I'm pretty excited about that. It's a really, really big number, and it means a lot of people care about Alzheimer's disease and are trying to help us find a treatment. Next. So on any given day, you have maybe 50 to 150 different people that are actually engaged in the activity. So we have some people who are there every day playing hours at a time. We have some people who stop in maybe once a week, and then we have people who come in actually once a year for events like this one. But everybody in total contributes, and because of everyone's involvement, we're getting the research done 5 times faster. Next. And because of that, we're able to answer these research questions about a prospective treatment every 1 to 2 months instead of every 6 to 12 months. So that's taking what potentially would have been several decades worth of work and kind of compressing it into a much smaller period of time. Next. So as Emma mentioned before, so we do publish our results in top tier journals, and when they let us, we will include stall catchers, which you can see on the scientific reports. One here says stall catchers there near the bottom, and then we'll hyperlink it to a list, so then you can see the actual usernames of everyone who contributed, and I think we have it set up to list those in the order in which people contributed to it. And these are some of these journals are journals that career scientists spend their entire careers trying to publish in. So it's kind of cool that someone who participates in Stall Catchers might be able to say, hey, I'm published in one of these top tier journals, and of course something we really want to recognize, because without people doing the activity, we wouldn't have these results. And the other kind of cool thing is that occasionally somebody like any of you here today might come up with a question, and we'll run with it. We actually had a catcher say, hey, what about the impact of exercise on Alzheimer's disease? Is that something that we might see through this capillary stalling phenomenon? So one of the graduate students in the Schaefer-Nishimura lab decided to run with that, and they actually did a study where they had some of the mice. They had 2 groups, both with Alzheimer's, but one group they put on a little mouse treadmill every day, and so that ended up becoming a full-fledged study with published results. So next slide, please.
Hey, sure. Any chance you remember the outcome of that study?
Yeah, I was kind
I'm
of
very
afraid you were going to
curious.
ask that question.
Oh
So
no, sorry.
I think no. It's a really good question. And if I remember right, the overall, I think the mice overall did better. They did better on cognitive performance tests. Their health overall was better, but I'm not sure it actually impacted the capillary stalling. So it was kind of a mixed finding. So it's like, yes, it's good to exercise if you have Alzheimer's, but it's not the mechanism necessarily that's going to make those stalls go away.
Interesting. That is super interesting. That's very cool.
And that's the I think for me the fascinating part about doing science is that you often have an idea of what the result is going to be, or you're hoping it's going to be a certain kind of a result, and sometimes it just turns out to be completely different than what you expect, and that's not necessarily bad news. Sometimes it's actually good news, because it keeps you from going down a path that wasn't really going to lead anywhere, and helps redirect you to a more fruitful kind of research path. So as I mentioned, if you're looking at this slide, the idea here is is that we can take drugs that we think could be potential treatments that would make these stalls go away, and we have, and by we I mean the Schaefer Nishimura lab. They've taken drugs that are being used today that are FDA approved to treat cancer because they had properties that they thought might also help reduce stalls. And what they found is that one of them actually does slightly reduce the stall rate, but it isn't clear that it reduces the stall rate enough that it would make sense, because, as you know, a lot of chemotherapeutic agents have side effect profiles that aren't great. So it's like one of these things that is kind of like, okay, feels like we're kind of on the right track, but we're not there yet. And sometimes they'll give the mice something that isn't a full-on drug or potential treatment, but something that'll just reveal something about the upstream or downstream molecular pathways, which is what you're seeing here, where it helps us get a better sense of how are these stalls forming in the first place. Is there some pathway we can interrupt there? Or once the stalls have formed, is there something we can do after the fact that would kind of, mitigate, make the stalls go away? So these green check marks are these very specific studies that have been done and research questions answered in stall catchers that helps us get a clearer picture. And some of these red question marks are data sets yet to be collected and analyzed in stall catchers. And this is an older slide. I think we actually have more green check marks than what you see here now.
I was actually going to ask about that because I think we pulled this image quite some time ago, so that's good to know.
Yeah, I should circle back with the lab and see what it looks like today.
Sweet. Looking forward to it. Oh, Caroline, hey. Sorry, I
Yeah,
wasn't sure if you were trying to.
no worries. I was gonna say maybe after today's event we can update this even more. Um, Yeah, but that's pretty much the blurb. I checked Facebook, and quite a few people are watching on Facebook as well. So I wanted to let our Facebook viewers know, if you have questions for Pietro, obviously don't hesitate to ask in the Facebook comments. We'll make sure those are relayed over Zoom. But I think we should go ahead and start catching, and then we can get into some Q&A after that, if that works for you, Emma.
Yeah, or we can mix it all together. So if you do have
Let's do
questions, we
it.
can just answer them as we go. So let's get you set up. So for those of you with us today, we're going to teach you how to get signed up. So I'm going to go ahead and visit the SciStarter, except of course I couldn't. Me. If I, yep, there it is. Okay. So I'm going to visit SciStarter/stall catchers, which is now an active link, as well as a couple other, oh, of course I spelled it wrong. Ah, and I'll drop this in the chat for you, or just kidding, Caroline already did. So when you go there, this just gives you a good breakdown. It's like a one-pager of the project. You can, of course, find all this information on stallcatchers.com as well. We just like to direct you here just so you can see what it looks like here, and so you can add it to your dashboard. So I'm going to go ahead and hit visit, which will just take me to the main site, assuming my internet catches up. There we go. And I can log in. So I already have a login. If you don't already have a login, you can also register. So I'll go ahead and link that in the chat as well. Register. And if you have a SciStarter account, if you create an account on Stallcatchers using your SciStarter email, so the email that you use for your account on SciStarter, that means that the accounts will be linked, so you can check on all your contributions through your SciStarter dashboard as well, which is really great because you get to just kind of check your progress over time, see how often you participate in different projects that are affiliates, which is what this connection is describing. And you can help us understand more about how to benefit the citizen science industry as well. So I'll go ahead and make up a name. Today is September 30th. December 30th, SS Live.
And while Emma picks
And
her new
then,
team, just wanted to remind everybody, because this always trips people up, it's two different accounts. You're just using the same email address to sign up for both. So you're doing just what Emma's doing. You're making a new email address, and theoretically, she also used the same email address to our new account with the same email address. And theoretically, she also used it to sign up for SciStarter some time ago. So if you don't have a SciStarter account, never a bad time to make one. You can do that on scistarter.org. And then once you have your SciStarter account, if you make your account on Stall Catchers with that same email address, the number and frequency of your contributions will track back to your SciStarter dashboard, which is great for bragging rights or to get different badges on SciStarter and all sorts of fun stuff.
awesome. Yep, precisely. And then luckily for stall catchers, you don't have to worry about a password yet. So that's just sent to your email for you to address later. So I just created a new account so that it would send me back to the beginning. Let me zoom in a bunch. And so it'll take you through a tour. So I'll do a brief tour. Feel free to interrupt me, Pietro or Caroline, if I miss something that you want to spend some time looking at. But this is the game. So game that speeds up Alzheimer's research. This is the interface. When you've signed in. Oh, I do want to make sure I'm not rushing ahead in case anyone wants some more time or has questions about sign up. So feel free to interrupt if you have any concerns or add into the chat so we can help you. Roland is at the ready to support. So it's a virtual microscope, which just means you're looking at things that are really tiny. So it has to be virtual and you're not looking at a microscope. And these are close-up movies of blood vessels in the brains of mice, as described earlier. You have the normal assumed items or menu items like a scroller to see the movie and decide where to start in the movie. You can move it around however you need to. And then there's a movie frame number for you as well. I actually use that mostly here to just ask where people see the stall. So I will be asking for you to find where you might see something. This is the most helpful information that I ever give people in my mind is that you're not looking at the whole image. You're looking specifically at this orange area that's outlined because there are going to be a bunch of blood vessels in this section, but you're only going to be looking for what's happening in the identified outlined area to decide if it's flowing or stalled. And then that's the big picture. We really need to know one of two things. Is it stalled or flowing? So if there are black spots, blood cells that are stuck and they do not move inside the vessel, it's probably stalled. If you find one, you can click the stalled button. I like to use the relation that it reminds me of a milkshake with chocolate chips. When a chocolate chip is too big and gets stuck in the straw, it's just a black chunk in the middle of the straw. That would be a stall, right? Hopefully you eventually get that chocolate chip eventually, but that's where the analogy kind of breaks. So for flowing, you might see black spots, but they are not stuck. It's just flowing. There's nothing stuck. There's just movement. The blood seems to be flowing in a single direction. The vessel is flowing. If you think so, click the flowing button. Anything to add to those two descriptions? My experts on the line.
I just want to say I love the milkshake metaphor, which
Thanks.
I've never heard before. At the very beginning of the project, when we had it at it was the Science and Technology Engineering kind of festival in Washington, D.C., with a ton of people coming through with families. We had a prototype. We didn't actually have stall catchers launched online yet, so we had a prototype, and kids would come by and we'd say it was zebras. So we had
Oh.
zebras with moving stripes were magical zebras, healthy zebras, because the blood's flowing. And then when the stripes were stuck and kind of like regular zebras, those were not the magical zebras. So anyway, I think the milkshake metaphor works better for me. I think that's good.
It took me at least a year to come up with that, so if that helps.
Wow. Okay.
Uh,
Impressive.
Emma, we need to
All
write
the
that down. That's a good metaphor.
yeah, for sure. I mean, I can recite it by I do it in all of the events for stall catchers now, so I'm glad to know that it's actually a workable metaphor. And there's also a help section for all of you who are looking at this screen on your site. You can always go back for more help. Yeah, we should definitely. I'll write a blog about it. Okay, so we're going to do some training videos first. So you'll first do about 10 that are just for training. So it'll gauge your ability to answer correctly. When I hit done, I'll be able to use the slider to look at the current image. So I'll go ahead and hit done so we can see this. Again, this is a new account, so everything is down to 0. I have a 0 score, level 0. My rank is kind of random, or at least I'm way at the bottom, right? And then I will be also able to see that I'm on Team Humanity. When you first join, I assume you'll all be on Team Humanity. I think that's the automatic. And so when you join a team, if you would like to, you are welcome to. You can always click choose team and it will send you elsewhere to pick a team. There is one for SciStarter. I'll go ahead and go back actually. Ooh. Oh, I hadn't started. So I can actually see what's happening today. So I'm going to go to my team. I'm going to say this week so I can see who's on my team. Oh no, that's going to be tough to beat. Star Rider on every single day. I'm pretty certain about that. Super dedicated. And then I'll just hit play to go automatically through the video and see what's happening in my little outlined area. And then as I start getting points, we'll be able to see what might change over to the left-hand side. All right, so first video, just off the top of your heads, how you feel about this video. Do we think it is stalled or flowing? If you have an answer, feel free to write in the chat while I sing the Jeopardy song. No. All right, we got one for stalled. Ooh, and the frame? Okay, let's see. Let me go to that frame.
This is the most stalled blood vessel to ever be stalled. But I mean, that makes
Does that
sense
mean
because it's the training video. I'm actually curious of the people in the chat. I know one person said they had done stall catchers before. Are there other folks who this isn't their first rodeo? Also, if it's your first time, like, don't worry. I know there are some people on Facebook who haven't done it before, so like, obviously, we'll go through the whole training with them, and they can also at any point redo the training on their own. But just checking the chat to see if anybody else is William's done it before. Okay. Any other old veterans of stall catchers in the chat? We'll let them
I
answer.
have
I also
never.
Yes, and before Emma clicks on the stall, agreed this one is nice and easy. Yes, definitely. Before she clicks on the stall, I thought it also might be worthwhile. So like once we make it through this training, and I think the training's super valuable. I redo it sometimes just to remind myself if I haven't done stall catchers in a while. Um, this particular data set that we're analyzing, so you see that in the top right-hand corner, the 44% data set analyzed. This is for a data set that will help stall catchers calibrate our data quality. Actually, Pedro, do you want to say anything else about that? I thought that might, because I know we have some people in here who are probably off to the races and already catching because they've done it before. But anything you want to say about the data set they're analyzing before Emma gets back to the training?
Yeah, sure. So this is a data set that was previously analyzed by the catcher community. And when we when we had folks at the lab analyze a recent data set, they said, something seems a little different to us, and we want to make sure that we can still trust the data coming out of stall catchers. Could you look into this? So one way to look into it is to say, all right, let's take, to see is stall catchers analyzing the data differently or the same as it always has. So we took a data set that was previously analyzed, and now we're reanalyzing it. And what we think we're seeing is that that there's been an overall shift in the bias of catchers. So what does that mean? Your bias is when you see a vessel and you're not sure if it's flowing or stalled, you're kind of in the middle. Are you going to lean more toward calling it stalled or more toward calling it flowing? Um, and in the early days of stall catchers, um, it seemed like there was pretty consistently over time, everyone kind of had the same, like, or at least the community had kind of the same average bias. And I don't remember if it was leaning more towards saying flowing or stalled, but it was pretty consistent over time. And what we're wondering is if that bias has shifted. And we actually, in the first few years of stall catchers, we had a mechanism to account and adjust for changing bias. And we actually did another test where we reanalyzed the data set using this control. And it didn't seem to make a difference. The results came out the same. And so we thought, well, if it's not helping, we don't want to introduce something new that might alter the results in any way. So we took it out. So if it turns out that it actually is making a difference today, then we have this same kind of solution already ready to deploy. If it turns out that the bias hasn't shifted and the results come out exactly the same as they did before, then it could be one of these research results that the Cornell lab got where it was just different than what they were expecting, but it happens to be the actual result. And then they have to kind of rethink, oh, okay, what does that mean? So I'm curious to see how it all plays out.
Ooh, we have a comment about this in the chat. I'm going to read. William says, it's really interesting. When I first learned, I leaned towards stall, and now I think I'm more likely to lean towards flowing. That's based on the expert answers I've seen while doing it. Yeah,
Oh,
I have a
that's
feeling I
very
was probably leaning
interesting.
towards stall too at the first starting point, and then as I got through more examples, I realized it's almost like we have a bias of assuming there will be fewer stalls in the data set than flowing answers. And so it's like a attempt to get the likelihood or like a probability thing in my brain.
That's actually very interesting and helpful. So I appreciate you sharing that. I think, there's one possibility is that, and especially hearing what you had to say as well, Emma, that newer catchers will lean more one way and adjust the other way over time. If that's true, then when we do an event that involves a lot of newcomers, it could be that during that event, the bias is actually shifted because they're still, kind of getting used to how to analyze the stalls. And that's something we can directly analyze through our data. We have so much data, and we can see for each person who's contributing how long they've been in the system, how many stalls they found, or how many vessels they've analyzed, and we can actually do this analysis and answer this question. So I think you just gave me a research question that I'm really excited to dig into now.
Oh, sweet. Make sure I'm listed as the co-author. No, I'm kidding.
Well, yeah, yeah, and also William. Yeah.
Yeah, and William.
I think that's
Plus
fair.
you
We should
learned that
make a note.
there,
Mhm.
yeah, and William also added another night, like a night note, excuse me.
Okay.
Plus, oops. Plus you learned that there are crossover vessels that look like a stall but aren't, so you learn to look for them. Ooh, crossover vessels.
Mhm.
I'm not sure I know what is happening there. I wish I had an example right now. Um, like where the vessels are like overlapping.
Well, so I mean, I'll let William answer, but okay, sounds like you got it right. Yeah.
And when that happens, they look like they're stalled, but they're not. Ooh, so maybe so I'll keep moving in the background too to see if we can get to something that looks similar to that. But if anyone gets to that while they're playing and online with us on Zoom, we can also share from other participants too, screens. Any note on that, Pietro? Have you seen that where that might be tricky?
It's a good question. Yeah, I think, anytime you have vessels that are crossing in front of each other or behind each other, I think that can, make it trickier. Another thing that can happen is that, if it's a broad outline like this one, you you might you might at first think that the vessel you're supposed to be looking at is a different one than the main vessel that follows the outline. And then if you analyze that vessel, maybe there's a stall in that one, but not the one that's intended. Um,
Hmm.
and I think one thing, we don't say this right off the bat because it's almost like it's easier to learn it perceptually than to have someone explain it to you. But the way that these videos work is, at the beginning of the video, you're kind of at a surface level of the area of the brain that you're looking at. And with each frame, you're not only moving through time, but you're moving successively down through layers of brain tissue. And that's why vessels come into and out of view over the course of the video, is you're actually kind of going down and taking this kind of cross section lower and lower in the brain. So, and as you can probably imagine, not all the vessels have decided to cooperate and be flat. Sometimes they're vertically, oriented. So these big white spots that you see, in, yeah, like that one, Emma, thank you. Yeah, those two white spots are, they're also fatter vessels. So they might be venules or arterioles, which is basically blood vessels that are bigger than capillaries. But it looks like they're oriented vertically, and that's why it's just a circle, because it's straight up and down, and you're just kind of like looking at it at different levels, if that makes sense.
Yeah, this reminds me of when there was a orange outline on one of those, and I think I was literally on the call with you demonstrating this for folks, and we were like, wait a second, that doesn't seem like it was meant to be highlighting that section, or at least that's my memory of it. So I think we even flagged that one. So on in demonstrating, we were able to identify where things can be
Right.
trickier than we expect.
Yeah, and this happens a lot. We'll get spurious outlines because the generation of the outlines is an automatic process. There are no humans drawing these outlines. The machine is going through and trying to identify these vessel segments, and sometimes it gets it wrong, and that's why we have the mechanism to flag it.
Right. Which, if that happens to any of you, it's right here. You
That's
can
not
always
sure.
flag it
Oh,
there.
there it is. Okay.
Oh, is
Yeah.
it not showing?
Now it is. Yeah.
Okay.
Maybe it's
Yeah,
just
I
a
think,
delay
I
on
mean,
my
I've
end.
only ever hit it once, that's for sure.
Hmm.
And of the many, many times I've participated. Um, it's funny, this is the one that always tricks me up, even though when I've done demonstrations of this, I always end up being on this question asking myself, I know the answer, but I think I make it an error every other time I do this one. So for those of you watching slash participating, what do you put for this answer? Is it flowing or stalled? Oh, and Cassandra earlier said, noticing the overlapping. If you do, yeah, Cassandra, if you notice one overlapping again, let me know and we can look at it. Awesome. Any thoughts? You might also be very focused on what
Yeah,
you're doing on your computer.
yeah, I know
Oh,
some
thanks,
people
William.
are actively catching, but I think I know William said stalled. I think this one's flowing. I'm with Cassandra on this one.
All right, majority rules would make it flowing. I do want to note that the reason this one trips me up is because of this section. It looks like it doesn't get lit up, but then there is a little bit of gray in there. So I get tricked every time. Let's see. Oh, phew. And then I get passively aggressive notes about my this this one is definitely flowing. I love it. So if you feel passively aggressively talked at by notes, you can feel my pain. William, thank you. Sorry. Okay. This one just, it's interesting how they differ from, is it data set to data set or like based on, I don't know, it seems like the quality or type of video almost changes. Like this one's slower moving and it's like a fuzzier image. I don't know if there's any reason for that or if it just kind of luck of the draw, but it is interesting to see that change every once in a while.
Yeah, there are all kinds of reasons for this happening. From one imaging session to the next, sometimes the researchers will experiment with different settings
Oh, okay.
just to see if they can get better images. We also saw with the high fat diet experiment that all the images were noisier, and that wasn't an imaging issue. It's because the brain looks that way when you're eating a lot of fat, which is kind of scary. So there was just
Oh
all
my
this
gosh.
extra noise in the image. I don't know if that's just lipids reflecting light. I'm not sure exactly how it works.
Oh,
So
interesting.
yeah, and early on we had a lot of motion artifacts. So these are live mice that are being imaged in vivo, meaning they're still alive. They have chronic plates installed in their skulls, so it's kind of like a glass window um, that allows you to see their brain. Once they're anesthetized when it's installed, once they recover from the surgery and they're no longer under anesthesia, they don't feel it. They don't even know it's there, so they're not suffering in any way from this. Um, and
Nice.
and so yeah, that's so then these mice will participate. They'll be imaged at one point in time, and then maybe weeks later they'll be re-imaged, so we see if there's kind of a cumulative effect and so on. I think I was going somewhere else with that, but now I kind of lost my my track.
No, but that is really, I've actually been asked a couple times, or at least once, about the use of lab animals using lab mice. And I'm glad to have an answer for that now, actually. Thank you for mentioning that. Because I was curious how this works if they are, I mean, I assumed they were
Yeah.
alive, but had no idea how that works. So,
Right. And so during the imaging sessions, they're sedated. Oh, I was talking about motion artifacts. So while they're being
oh,
imaged, their heart is beating and they're breathing. So they're in a very sedated state, kind of like, when someone goes in for a colonoscopy, you don't remember it afterwards. Um, but but they're still alive, so they have biological motion in their bodies. And so we were getting a lot of weird artifacts in the videos from that. And then we had a member of the lab, Mohammed Hof Jafaharian, who went to Princeton to spend a week there one summer with the folks who were part of a different citizen science project called iWire. And he was trying to understand the algorithms that they were using and if they could be applied to help us reduce these motion artifacts. and it turned out to be quite successful. He came back and he adapted the algorithm, and then we applied it, and then all of the weird kind of heartbeat and breathing motion went away. It stabilized the images. So it was remarkable,
Wow.
actually. Yeah.
Huh. What was the sorry, what was the magic trick that worked?
It's just it was kind of a computer vision algorithm that had been developed at
Um,
Princeton, and then excuse me, but they do imaging on neurons there, and Mohammed adapted it to work for these kinds of images.
oh, that's awesome. See, you're for participants in stall catchers, this is like a you're entering a world of enormous amounts of research that, I mean, it taking decades to get through individual questions and us compressing that is this is incredible. Like it's really this is probably one of the prime examples of citizen science allowing for that ecosystem to move so quickly. Uh, yeah. If anyone has an idea about this one, what do we think? It's nice and light. Scrolling through. Looks like magical zebra to me. Uh,
Ooh,
movement.
maybe let's play it on a loop for a little bit, just so we can kind of
Sure.
contemplate it. I was also going to say, I know we have 12 minutes left together, so everyone feel free to start catching like mad, or you can just, for those who are still getting oriented, you can keep watching and following along with Emma. But you know, the month ends at midnight in your time zone, so and also you can do stall catchers anytime. But if you're doing it to commemorate Alzheimer's Awareness Month, you still have a little bit more month left, if you want to keep going. I think this one's flowing. What do you think, Emma?
I
What
agree.
William agrees
And
with
William
me.
agrees with us too.
Yes.
So I am gonna go with that. Yes. You're doing great. Keep going. Woohoo. All right. So
Woo!
our team of presenters, let's like do a set of five very quickly. See if we can get consensus. Although this is training one, so Pietro and Caroline and I probably have the actual answers, but we'll still do it anyway. Um, all right. Looking at this one, my inclination is stalled.
I think stalled too. Wait, let me be. Yeah, stalled. I would say stalled.
Okay. Are you looking at this section?
Yeah, and the bottom quadrant. Yeah,
Awesome. Same. Let's see how we do. Yes. Estelle is clearly visible. Sorry, I like the wording
yeah.
for
You
the
can
sake
tell
of
Emma and
making
I
the
have
joke to
had
people.
a lot of stall catchers practice over the years.
Oh yeah. Oh yeah. And 70 points later. Woohoo.
Oh, did we
I
mention
can jump
the chat
actually.
yet?
Sorry?
We haven't mentioned the stall catchers chat yet, right? I feel like that's a great
Oh,
resource
no,
for
we haven't.
people.
Ah, over here. Oh, maybe it's because my screen is too
Yeah, you
close.
zoomed in for the sake of Zoom, which makes sense.
But actually I think my thing isn't fully loading. Let's see. Oh, that's odd. The chat usually shows up here, so maybe it's because I haven't done the question. That's not
That
supposed to
makes
happen,
sense.
right?
Well, that one's stalled, so we know that.
There we go. Okay. Oh, yeah. Okay. For some reason, it didn't want to. There we go. There it is. Oh, hey, Herbialdo. He's on the call with us. Hi.
Yay!
And then there's this also lovely thing.
Yeah, I
Hello,
find that to be a really good
SciStarter.
resource, especially if you have a particular grievance against a video. You can make your voice heard, not in the daily online chat. That's more just to correspond with like, some of the like Caparom, he's like a super catcher who's always on there, Mike. So you can like, and I see a hello from SciStarter from Emma, for example. So that's more fun for like daily correspondence, but there, what you saw earlier, the forum underneath the main video, that's useful if you have like a grievance to air where you're just like, there was definitely a stall at this particular frame, that kind of thing. And you can always post that there.
I really want to know the details about how often Star Rider participates, so I'm going to ask
Hey, that's the
how many
purpose of
videos.
having, a chat so you can correspond with each other.
Yeah, I feel like I'm in league with superstars because we do, whenever I see these folks, like the top five, they're just, I mean, they do such um, incredible amounts of analysis. And this is, sorry, I have like 15 billion things open, so I think my Wi-Fi is stalling. Hopefully you can still hear me.
Yep, we can still hear you
Let's see.
loud and clear. While Emma
Okay.
classifies this next video, I also wanted to flag Pietro just put in the chat. I know Roland's transferring it to Facebook, a link to the leaderboard. So, like I said, month's not over yet. Depending on where you are in the world, you have a few hours. You can continue to classify and get on up in the leaderboard or see some of the awesome people who have already and get inspired by them. Like Emma mentioned, the superstars of stall catchers and see how they're doing on the leaderboard through the end of this challenge. The 17 hours or so that might remain. And of course, you can do stall catchers anytime. And if any of you, like, I know a lot of the awesome people on the line have access to like cool communities, like maybe you're head of a Girl Scouts troop, or maybe your coworkers at Verizon would just love this, or whatever it might be. You can always get a team going for your unique community, or if you have like a whole big group of people and you want to do different teams, you can have a league. But we encourage you to do stall catchers with your friends, family, colleagues, and we're here to help you do that.
Absolutely. I figured I would reset it with my, oh, and yeah, it's working. Reset it with my live account so we can see ones that are not just in the training. So for those of you who pushed forward, you might be seeing ones like this, which actually beautiful one to start with, because I do want to ask the question of these ones are very challenging, like how to gauge what is happening in here. So Pietro, help me out. What's happening? What's your advice?
I don't know. This looks like a stormy sea to me. Um, uh, yeah, I don't know if this is, it looks like the high fat diet data set a bit. I would flag this one. I don't. I feel like I wouldn't be able to analyze this, and I'm not sure that that's actually surrounding something that's interpretable. So I would,
Mhm.
when you flag it, you just turn on that red flag, and then it doesn't matter whether you pick flowing or stalled at that point. we
So
still we
I'll hit
still ask
flowing so it
people to
doesn't
give
ask.
give their best guess, no matter what. But
There we go.
one thing that some people might find interesting, Emma, is if you scroll up to the top, there's um, you see where it says Emma G there, your
Mhm.
profile menu. So if you click on that, there's a timesheet option. So some people who are trying to, keep track of how much time they've spent on it. Some people do it for volunteer hours or for other reasons, or some people just want to know, I've been playing stall catchers for a year. How much time have I spent on it? You can choose whatever period of time you want, and you can get you can even print it out. So I think it'll generate, kind of an
Ooh,
official record of it. So if we get students who sometimes say, hey, I'm doing community hours for school. Can you certify that I've done this? And then we just point them to this, and then this serves as an official record.
that's awesome. I'm so glad I know now where to find that because I've gotten that question before.
Oh,
And yeah,
okay,
and I
good.
love that stall catchers definitely the whole idea of being able to translate it to hours is pretty unique for projects. There's very few that can do that so distinctly, and so it really is a very nice thing to have available. So yeah, definitely would recommend stall catchers to volunteer for students who need to volunteer and prove it
Hmm.
and prove it. If I go back to, actually, let me go to, no, not leaderboards. I was going to go to my team. Oops. Let me go back to catch. I'm in a different team right now, I think, all Verizon squads. So let me switch back to my SciStarter team. And then we'll go ahead and close this out anyway, because we're getting close to the hour, about 5 minutes left. If I have time for another one of those, I believe. Oh, doesn't want to show up. Maybe I got rid of it.
This
All right,
might be,
I will
if you have
join
any
my
final questions, go ahead and get those in the chat as Emma gets set up to close this out.
just go back to these. Okay. Star Rider just says a bit of practice for how to get to the scores that they have. Dang. Could have guessed. Awesome. And they're currently at 4 million. Incredible. All right. This one does not have as much noise as the previous one, so hopefully, let's see. I'm tempted to say, ooh, I'm tempted to say stalled, but this is like looks like a circular, like I'm looking at this blood vessel, I think. Does anybody else have an opinion on the line as our final tough one? Oh, or wait, it's that one. Sometimes the outline is tough to decide what is the primary vessel
Emma,
to
you
look
might
at.
not be sharing your screen. Or wait, maybe I changed my view.
Am I not?
Oh,
Oh,
yes, you are. You are. I lied.
okay. It's like, that's awkward.
Yeah. I think I agree with William on this one. I
Ooh.
think I would, I'm wrong all the time on these, but I also think it's stalled.
Do we think it's stalled here, like in this section, or before?
I think it's the earlier one is the vessel that goes around in the circle more. Yeah, I
This
think,
area?
but yeah, that's what I would go with.
Okay, yeah, it's really dark there and then lightens up in some areas, but doesn't look like it covers all of it. We'll do that.
And level up.
Yay, thanks team. This is unfortunately, or not unfortunately, it's fortunate for me, but since I love doing this with volunteer groups, I make them do all the work and then I end up with crazy amounts of points by the end of the So I'm just cheating a little bit. But it's okay. It's the power of the crowd. Thank you, William, for your help there. All right. And with our last couple of minutes left, any questions for the team? Any comments or quotes about how much you love SciStarter? I will put you on the internet on a social media post. And we're really grateful that you can all join us.
Woo!
All right. Well done, team. Keep going till midnight, wherever that is in your time zone.
I guess
We
I'll end it with one last question for Pietro. For Pietro, do you want to tell people maybe what's next for stall catchers and the Human Computation Institute?
Um, yeah, I don't know if I can say that in 1 minute, but we do have another project that's becoming fairly mature. We haven't publicly launched it yet. It's called Beta Catchers, and it's also to help advance Alzheimer's disease research. But what you're actually doing in the activity is quite different because you're looking at slides, not videos, but static slides of human brain tissue from deceased Alzheimer's patients, and you're trying to locate actual amyloid plaques and other pathologies in the brain. So you're taught what these things look like through a tutorial, and you're doing the work of a trained pathologist by the time you're actually analyzing all these things. I think I actually find it kind of fun. I feel like, as with most gamified things, some people gravitate more to one type than the other, but I think this will appeal to some group of people, and we'll definitely let you know when it's gone live, and you can give it a try.
So exciting. I'm very excited about that one. I feel like every time I find like some projects just make you feel like a true scientist, and so your commentary about doing actual pathology work is very memorable to me. So yeah, get ready to be a pathologist, everyone.
Exactly.
Awesome. All
Thanks,
right,
thanks
go
everyone.
team. Thank you
Really
so much.
appreciate it.
I'm just gonna drop this in the last couple seconds in the screen in case you need any support from us. You are always welcome to reach out to us. You can request different events for different projects. If you would like to see us do something else next, feel free to let us know. We are happy to oblige and help you use SciStar to the fullest extent and answer questions about any of these projects, including stall catchers. So feel free to reach out and we will see you next time. Thank you, everyone.
And thank you, Emma, Caroline, Roland, for hosting
Likewise.
this. Yeah, appreciate it. Thanks, everyone.
Thanks for joining us. Appreciate it.
Yeah.
It's always fun.